Cell transportation process means the letting in and out of liquid, gases, or solids. It is divided on passive and active. Passive means that you don't need any energy for your molecules to pass through, just like the oxygen. But active means that you have to spend some energy for your molecules to pass through, just like proteins and ATP. Hypotonic solution is defined by a higher concentration of water and a lower concentration of solute, where the water is transported into the cell, and the cells inflate and eventually burst. Hypertonic solution is defined by a higher concentration of solute and a lower concentration of water, where the water is transported out from the cell and the cells shrink. One of the principal characteristics that really define animal cells is that compared to plant cells, they are quite smaller and that their DNA is housed within the nucleus. The molecules that can pass freely through the cell membrane are oxygen and carbon dioxide. So right now I'm gonna explain to you my step-by-step project. So number one is, I started my process with 2 eggs. So I took one flask and I took the 2 eggs and I put them there and covered them completely with vinegar. And I left them there for about 3 days. The purpose of this step was to get rid of the hard calcium layer that was surrounding the egg, because we need to get rid of that so we can work with them and show our process. After 3 days that I let them in the vinegar, I removed them and I rinsed them with water. So now the eggs have been exposed to a semi-permeable membrane. I weighed them and my first egg was about 0.83 and my second egg was about 0.84. After that, I needed 2 flasks. One covered completely with water and the second one completely with corn-sided. So I took my egg and I put one on the water bottle and I put the other one on the corn-sided. The water one is supposed to be the hypotonic solution one, and the corn-sided one is supposed to be the hypertonic solution one. I left them there for about 8 hours, and I've started noticing that there were some changes happening with them. So I just weighed my first egg and it's about 0.88 and my second egg is about 0.78. On my personal conclusions, I can say that in the flask 1, with the hypotonic solution, the water got like that, because the hypotonic solution is defined by a higher concentration of water and a lower concentration of solute. So the water is swollen by the egg. So that's why the egg got really big. Meanwhile, with the hypertonic solution is defined by a lower concentration of water and a higher concentration of solute. So that's why the egg got shrunk, because it didn't handle any water, because the water get rid of. When a cell is placed in hypotonic solution, it gets really big because the water is swollen, and the concentration of solute, it gets really low. A cell placed on the hypertonic solution, it gets swollen, it gets tiny because the concentration of water gets really low and the concentration of solute gets really high. So osmosis is a type of cell transportation process, which is a passive one, because it doesn't really need energy to produce it. It's divided on hypertonic solution, which is a higher concentration of solute and a lower concentration of water, a hypotonic solution, which is a higher concentration of water and a lower concentration of solute, and the isotonic solution, which is the same level of water and the same level of solute.